Welcome to Lecture DS-2, the second lecture of the digestive system. We're going to start this with a quick overview of some of the enzymes involved in digestion and a little bit about those. We'll then talk about digestion in three phases. We'll begin with the cephalic phase, which will involve what goes on in the head and neck area. We'll spend some time talking about saliva and swallowing. We'll then get into the gastric phase, which is what goes on in the stomach. We'll talk about the gastric glands and the hormones and secretions they make, including stomach acid and pepsin and intrinsic factor. Then we'll get into the intestinal phase, where we talk about the responses of the duodenum as stomach contents enter it, how it interacts with the rest of the body and the digestive system. We'll talk about the secretion of inactive enzymes called zymogens from the pancreas and how they get activated, and the functions of brush border enzymes in the duodenum as well. And then finally, we'll talk a little bit about the absorption of different nutrients. Hi! So today we're going to be talking about some of the different phases of the digestive process in more detail than we did last time. We're going to be talking about three different phases. We'll talk about the cephalic phase, which starts as soon as you smell or taste or even think about food, and goes until the food reaches your stomach. We'll then talk about the gastric phase, which is what goes on specifically in the stomach, and how the gastric glands there in the stomach secrete various materials that not only affect the food, but also affect the rest of the digestive system and the rest of the body. And then we'll talk about the intestinal phase. After the materials leave the stomach, what happens to them then? How do we continue digestion, and how do we absorb the remaining materials? And then finally, we will talk a little bit about that absorption. Before we do that, we should spend a little bit of time briefly going back over some ideas with enzymes and giving you a few pieces of specific information. So, remember the idea of an enzyme. It's a protein which catalyzes a particular reaction. And in digestion, that's usually going to be breaking the bonds in those large molecules so that we can break them into pieces small enough to be absorbed. So this is, we're going to be generally breaking bonds, going from the larger biomolecules and breaking them into smaller pieces. For the ones that are polymers, usually that involves breaking them into their monomers. So proteins become amino acids, and nucleic acids become nucleotides, and all that stuff you learned about. Now, a couple of things about these enzymes. First of all, remember that enzymes are proteins, so their function depends on their shape, and their shape depends on how the amino acids interact with each other, which depends on what kind of charge they have, among other things. And that charge depends on pH. So, depending on the pH, proteins will fold or unfold differently. If you've ever had ceviche, you know that ceviche is acid-cooked fish. The acid cooking refers to the fact that by putting it into an acidic solution, you change the pH, which changes the way the proteins in the fish meat fold up, which is kind of like cooking with heat. It's just a different form of what we call denaturing the proteins. So that word denaturing means to unfold a protein, which causes it to change or lose its function. Now, the overall upshot of that is that if I start with an enzyme, a protein folded in a particular way, and I change the pH around it a lot, that can cause that enzyme to unfold and lose its function as an enzyme. Each enzyme will actually have a pH at which it works best. That's the pH where it folds up correctly. And if you change the pH very much from that, then it unfolds, it denatures, and doesn't work the same way. Given that the pH changes a lot as we go through the digestive system, an enzyme which works in one area may not work in another area. So enzymes get deactivated, and sometimes if you denature a protein, you can't fold it back up the right way. Sometimes denaturation is permanent. So you'll see a few examples of that. Now, I also wanted to go through some of the specific kinds of enzymes so that the words aren't new to you as we do this. So, in digestion, there's several specific enzymes we're going to work with. Amylases. Amylases work on starch. Remember, starch is a glucose polymer. And amylases take starch and break it up into maltose units. Starch is many glucoses together. Amylases will break maltose up into the disaccharide, so we'll break starch up into the disaccharide maltose, which is two glucoses together. Amylases can't break that down any further. The breaking up maltose has to happen later at a particular place in the intestines that we'll get to with a different enzyme. But that's the general idea of an amylase. A lipase is an enzyme which digests fat. And fat in foods is mostly in the form of triglycerides. So lipases take triglycerides, which if you remember is a glycerol and 3 fatty acids, and break it into fatty acids and monoglycerides. So that takes a triglyceride and breaks it up into a monoglyceride, glycerol plus 1 fatty acid, plus the other 2 fatty acids free. Nucleases break nucleic acids, like DNA and RNA, into separate nucleotides. So there's 3 enzymes: amylases, which digest starches into maltoses; lipases, which digest triglycerides into fatty acids and monoglycerides; and nucleases, which break up DNA and RNA into separate nucleotides. Now, the last enzyme is the idea of a peptidase. And as you might guess from the name, peptidases break up proteins. So you remember, a protein is a folded up chain of amino acids. In order to digest that, usually what we're going to have to do is denature it somewhat. That's going to be partially done with changes in pH. And then peptidases will attack this. And there's really 2 kinds of peptidases. Endopeptidases. Notice the name, endo, inside. Endopeptidases will cut this amino acid chain somewhere in the middle. So you end up with separate pieces. So endopeptidases will chop up that long chain into much smaller chains. And then exopeptidases will attack the ends of those chains, breaking them up into groups of 1 to 3 amino acids. Now, take a look at something on this one. So imagine that we've got a long, unfolded amino acid chain. Now you might say, why do we need an endopeptidase? That's got ends. Can't we just let an exopeptidase come along and chew through that and break it up along the way? We could, but that would take kind of a long time. Remember the idea of physical digestion, mechanical digestion, where we wanted to take a food piece and break it into smaller pieces so we had more surface area for the enzymes to work on? This is kind of the same idea. In this, exopeptidases only have 2 places where they can work, that end and that end. But if you start with an endopeptidase and break this up into lots of smaller chains, now look at all the places where exopeptidases can work. Here, 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 and here. We've made a lot more ends for them to chew on, which is kind of like the idea of increasing surface area. So the reason we have both endo and exopeptidases is that for the endopeptidases to increase the surface area, the number of ends in the protein chain, so the exopeptidases can work more efficiently. Alrighty, so there's our major enzymes. Now, our next thing we're going to do is talk about the cephalic phase, so starting with just even thinking about food and why your body responds to that and what kind of responses you get. Let's start by talking about the cephalic phase of digestion. So, cephalic comes from the root word talking about head, like my favorite creatures in the world, cephalopods, octopus and squid. Cephalopod means head-footed. So people thought that a cephalopod looked like it had its head just sitting right on its feet, all those arms. So the cephalic phase is the stuff that goes on in your head as part of digestion. This begins with sensory or cognitive aspects of food. So, the fact that I am smelling or seeing or even just a little taste of food, or even just thinking about food can trigger this. What this first part triggers is activity in the parasympathetic nervous system, which travels down the vagus nerve to the stomach. This is what is sometimes called the long reflex in the digestive process. So when I think about a tuna melt, my brain sends parasympathetic input down the vagus nerve all the way to my stomach and my upper abdomen. And that input causes the stomach to increase motility and secretion. So the stomach starts to add enzymes and acid into its lumen and starts to move around a little bit. This is actually, if you smell food and you're hungry and your stomach goes, that's actually what this is. The stomach is starting to move around, which you get a little movement of gas bubbles in there, and you get tummy rumbles when you're hungry. It's kind of fun. Anyway, so that's the first part of this. The stomach starts getting ready for food. And then we actually take the food into our mouth. So food into the oral cavity. And we begin the whole digestive process of the food actually coming in. So that's going to involve chewing, which is using the teeth and then using the tongue and cheeks and lips to move stuff around so that you can use the teeth to chop, grind, and squish this food into more of a paste rather than individual pieces. That's part of that mechanical digestion process, which is, in this case, is going to be about making the food soft and easy to swallow, and also increasing the surface area. The other thing that goes on here in your mouth is you add saliva. Spit. Saliva is kind of interesting stuff. Saliva is a liquid produced by the salivary glands. You have several sets of salivary glands around your mouth. And it contains a number of interesting things. Mostly it's water. There's some mucus in saliva. That water and mucus is mostly there to moisten and lubricate the food, to thin it out and make it easier to squish down into a paste and make it kind of slippery, so it'll be easier to swallow. Also, in your saliva is lysozyme, which is an enzyme for attacking bacterial cell walls, sort of a general disinfecting enzyme. So this lysozyme is one of your first defensive mechanisms. When you bring in stuff into your oral cavity, it's going to have bacteria on it. Lysozyme helps to start disinfecting that, to try to kill some of those bacteria. This also seems to be part of what helps prevent tooth decay. And you might know somebody, I'm one of those who just doesn't seem to get cavities. Last I heard, there was some evidence that that's not necessarily because of great dental hygiene, but possibly because some people have much more effective lysozyme that's good at killing those bacteria that would otherwise cause tooth decay. So, kind of an interesting point there. Anyway, we also have immunoglobulin A. I'll just say immunoglobulin here. Antibodies of a type that will latch onto these to microorganisms in the food and make it harder for them to get through the cell membranes in your digestive tract, harder for them to get into your body. In addition to all those, we have a couple of enzymes. I'll make a note that this is salivary amylase. So remember, an amylase is an enzyme which digests starch. So in your saliva, you have an amylase which will take starch molecules and start to break them up, trying to break them up into smaller pieces. Salivary amylase works pretty well. It starts the starch digestion process. It's not going to complete it. Starches here in your mouth do not get completely broken down into maltose, but it starts the process. We also have something called lingual lipase, a fat digesting enzyme. However, lingual lipase, as it's secreted in the mouth, doesn't actually fold up correctly. The mouth is too alkaline. The mouth is usually at a pH not too far from water, 6-ish. And lingual lipase doesn't fold up right until you get down to a more acidic pH. So here in the mouth, the lingual lipase doesn't actually do very much. Kind of interesting. Anyway, so this saliva is this complex mixture, and it starts to disinfect and moisten, lubricate, and even digest some of the starch here in your mouth. So you chew your food for a while and mix it with the saliva. And then it's time to swallow it. So swallowing involves using your tongue to shape that chewed food into what we call a bolus, which just means basically a sort of slightly rounded blob, and move that to the back of your, so move that into your pharynx. So as it goes to the back of the mouth, you trigger a series of muscular contractions, the swallowing reflex. The fancy word for swallowing is deglutition. Kind of neat. Anyway, so we move that bolus into the back of the mouth, and we trigger this series of contractions which move the larynx kind of up and the epiglottis down, so that closes off the opening to the larynx so that the swallowed food won't go there. So the food goes down past that, over the epiglottis, into the first part of the esophagus, that muscular tube from the laryngopharynx to the stomach. And there, the first part is skeletal muscle, and it then transitions to smooth muscle as we go. We start a peristaltic contraction, whereas the food gets in there, we start a wave of contraction behind the food, the bolus, that squeezes it down the tube toward the stomach. So if you feel your throat while you swallow, you can feel that rise as we're closing off the larynx. The other fun thing about that is once you start the swallowing reflex, it's very hard to stop it because you can initiate it, but once you start it, it's really a continuous process. It's not really under your control anymore, which is probably good because as that food's going down, you don't want to do anything that might open the larynx up and let the food in there. So it's better to leave that in the capable hands of your reflex. Anyway, so we swallow that, the food gets squeezed down the esophagus, down to that lower esophageal sphincter, that cardiac sphincter, which doesn't actually work all that well, but which then opens and the food gets squeezed down into the stomach, where we will start the next phase of digestion, the gastric phase.